Katharina Kopp. I started as a veterinarian here in Munich. I studied veterinary medicine in Munich. Then I did my dissertation here in the Tropical Institute in Munich on tropical ticks. After that I studied in Berlin, and this was tropical animal health management. And then I went to Africa and I specialized into molecular virology of zoonosis. Zoonosis, this means diseases which are in animals and can do a species spillover to humans and then are infecting humans. The most famous examples are HIV, which came originally from chimpanzees, or Ebola, which might be in a bat, but it's not 100% sure yet. But for Marburg virus, for example, the cousin of Ebola, it is very clear. We have found it in fruit bats in Uganda, and that's where I mainly work now. I'm doing virus discovery and developing field diagnostics for the discovery or identification of the natural reservoir of virus hosts. So what I'm actually doing is I'm sampling dead animals, wildlife animals, because we want to make very low impact on the wildlife. We don't want to kill endangered species for our research. So I work a lot with the community. I ask the community about their traditional knowledge, what they might have observed. Then we collect dead animals which have died of a natural cause, and then we extract DNA or RNA. Most of the viruses I work with are RNA, and my approach was a real-time PCR to actually detect the viruses. But I also work a lot with nanopore minion sequencing in the field. This is a real virus or pathogen discovery where I actually don't know what I'm looking for, but I just take all the sequences which are in there, and then that's why I also like this two ladies. I have later on studied bioinformatics also here in the TUM and LMU in Munich. And that is very handy when it comes to the metagenomics approach, because I have a lot of sequences and then I actually have to find out what pathogens are in there. So yeah, that's mainly what I'm doing, and why I'm interested in bio.kitchen is because now I'm an independent scientist. Before I was with TUM. Later on I was sent by the Australian CSIRO Animal Health Laboratory to Uganda. But at the moment I'm an independent researcher, and as an independent researcher I wanted to develop more fieldable assays. Like I would like to do real-time PCR in the field. I had a little real-time machine, but the problem is you always need power and it's very sensitive. And their approaches with microfluidics in real-time PCR and also isothermal methods or combining RPA with CRISPR, and there are a lot of new things now. And I'm quite confident and familiar with the wet lab side of things, but then I also would need some medical engineering, and I thought this interdisciplinary approach in bio.kitchen, and that it's an open community, so things are not in the first attempt commercial but open source, and that's very important for Africa because I also want to do technology transfer and things which are like cheap and open source, so the people in Africa can be trained actually also to design and build their own things. And that's why I'm also very attracted by the spirit of the biohacking and community lab and open source approach of things. And a very practical issue, I had bought a little bit of machines and equipment from eBay. These things are now in Uganda, like a very old conventional PCR and a gel illuminator and things like that. So this was no problem. You can get all these things from eBay. But when it went to the reagents, I wanted to, I mix my own master mixes because that's cheaper for the PCR, for the real-time PCR, but I couldn't get cyber green. The company said it's not allowed to ship me such a really, really dangerous chemical, which actually not dangerous at all, to my home address. Even I'm a single woman enterprise now. I founded this in German it's called Freiberufler, a little business, but only if you are an accredited lab, you can actually order lab reagents. They just don't deliver to a private lab. And it's also more convenient instead of like fumbling around in your own basement. And the community exchange here. There are always people who have just done something else and like iGEM and there are so many ideas, and I try to keep up, but then there are always new things, and I also found it not so inspiring to work on my own all the time. So I'm looking for a community. I'm very practically looking for a lab and a lab address where I can order reagents. And I'm also looking for exchange of like-minded people.
Would you maybe be interested to tell where the people might reach you? Because it's just amazing what you do.
Yeah, thank you. Thank you very much.
It sounds awesome.
This little company I founded is like a few weeks old. I have a Twitter account where next week on Monday I start. There's a real scientist affiliate in Germany. This is a bit of SciComm. I'm talking about what I've done in my recent research in Uganda and that. And then I've started, but it's in its infancy, a website. And it's a very complicated name. It's www.one-health-pathogenomics-consulting.org.
Okay, we'll write it down.
That's the website, and that's where you can follow up. There is a very practical thing. That's why people follow me on Twitter. A lot of people in the virus discovery community looking for novel zoonotic viruses. So what that means is like only a really new species or a very outlier strain of a new known species comes to the list, and it has to be zoonotic. So it means infectious coming from animals, infectious for people. Has to be novel. This virus species or strain has to be completely new, not known before, and it has to have available the whole genome, or at least most of the genome has to be available as sequence in the gene bank. Mm-hmm. And these are the criteria: zoonotic, novel virus, species, or at least strain, and mainly the main genes or even the whole genome of the virus is available. And then it comes on a literature list, and I try to keep it update every day. That's my morning procedure. And I have also, of course, I have a Twitter bot and everything, and like it's fully automatic, but it's curated because a lot of crap comes in there. So and from the beginning of 2018, I have already a list of like a bit more than 30 papers with novel zoonotic viruses. So you can imagine if 30 papers, peer-reviewed journal papers with novel viruses from animals which have been sequenced, that means the virus discovery at the moment is really exploding.
Yeah.
It might have been, it's an ongoing process, but we also talk about the emerging and new and emerging or re-emerging viruses, partially blamed on the climate change, but also like deforestation, encroachment, people walking or doing their farming in national parks, where actually before the rainforest mainly separated animal wildlife diseases from livestock, domestic animal diseases, and people. So there was this natural barrier. Maybe some Gaza hunters still living a traditional life are actually living in the rainforest, and we know that a lot of people, for example, are seropositive for Ebola, but these are hunter-gatherer indigenous people who are kind of adapted to this lifestyle. But when a person who is totally naive against these viruses is attacked by it, it's usually really bad because the virus didn't have time to mutate and adapt to the people. So we are kind of new. We are actually wrong hosts.
Ah, okay.
That's why it's so deadly. All other diseases which
So
have
there's a
adapted.
missing co-evolution.
Yeah, exactly.
Yeah,
Exactly.
okay.
Exactly. It's a spillover. That's why we call it spillover. It's like an accidentally happening jump into a new species, and
Okay.
that's why it kills so fast. Because other viruses, even influenza is quite deadly, but not so deadly compared to Ebola or something, because it had time to adapt to the people and people to adapt back. So and a lot of these environmental factors, that's also what I'm really interested in. I do a lot of community work in Africa. Before I have this a little bit of anthropological approach. I have questionnaires for the people and I do a survey because I think we can learn a lot. It's not always just we coming with our high technology as Europeans or Americans or other Western countries and to tell them what to do and how to do it, but they have lived with diseases like Ebola for decades, hundreds of years, thousands of years, and though there is traditional knowledge, like for example I have questionnaires, what do you actually think is dangerous in this forest? Which animals do you think are dangerous? Which animals are a taboo for you to eat? Things like that can be an indicator of the traditional knowledge the people already have. Because for example I learned that in Kenya, in the coastal area of Kenya, there are certain areas which are considered sacred. You are not, as a normal human being, if you are not the witch doctor or the traditional healer, you're not supposed to walk into that area. Otherwise you die. It's cursed. So they're like, okay, as a scientist, this is a bit questionable. But in the end, it turned out that these areas, which were degazetted for entry for people, were very, very deep into some small scattered rainforest, and there were caves inhabited by bats. And there were children, everybody was told, if you go there, you die. So these traditional knowledge, as stupid as it seems at a first glance, actually there is some experience behind it. It's not scientific knowledge, but it's knowledge by learning and doing and trial and error.
Yeah.
So I try to incorporate this anthropological approach by asking the people about their traditional African knowledge, and then I also get like leads or clues where to search. Because of course brute force, like in bioinformatics, brute force would be. I have a friend who is an astrophysicist who worked also in bioinformatics before, and he said, yeah, well, brute force, you should actually take all the animals of Uganda, put them in a mixer, extract the DNA and RNA, and then you will actually find your viruses. But you can't do that for some reason, and so we are still actually, after 40 years of like the first Ebola virus was not the Ebola virus as we know it from West Africa or Democratic Republic of Congo, but it's called Sudan Ebola virus. The very first of the 5 species of Ebola was Sudan Ebola virus. It was discovered 1976 in Sudan in a factory. People in a factory got sick where they produced cotton or stored cotton. And in this office of the factory, there were bats. So yeah, there's always this hint on that, but nobody ever had isolated.
Yeah. Do you want to join our discussion group?
Yeah.
Just come over.
So nobody has ever isolated or extracted Ebola virus from a bat. Not one of the 5 species. We have done that with Marburg virus. We know it's in fruit bats. For Ebola, the search is still going on after 40 years. So people are searching, I'm searching, others are searching, but we haven't found it yet. So we think we need very innovative new approaches and all kind of like science together.
Hopefully a lot of young scientists will see this, maybe even future iGEM teams looking for a new project.
Okay.
What are your ideas? Where should they start digging? Do you have some inspirations where they should look into, or what they could go for to tackle in order to help your idea and improve this?
Yes. What I'm actually looking for is a bit of like medical engineering. So we have the real-time PCR, and also we have some kind of sequencing like the Minion. What I would ideally looking for is a real-time PCR which is completely usable in the field. Like the Minion, like you need maybe a laptop, but you're independent from grid power, like something like with a battery pack or with a small solar. Because in Africa there is no power, even in the lab sometimes you don't have power, and then it has to be robust. It's a very harsh environment out there. There is dust, there is humidity, there is high temperature, and we call it like ruggedize, to make something very sturdy and robust. There are these approaches like from biodefense. I mean, for military, they have these testing machines which are incredibly expensive, and they are restricted because it's military grade research. So ideally, I want it open source. I want it for the community. I know Unternehmertum, but I personally would like to commercialize it. I would like it open source and community at least, or maybe commercialize it here in the Western. But make it approachable for the third world.